So today we're going to start to consider one of the two types of cell division, which is mitosis. The importance of mitosis cell division, or the significance of it, is that it has the ability to produce genetically identical cells, and it maintains the chromosome number. So if we consider a cell to start with, we call the cell that's about to divide a parent cell, and we have to know about mitosis cell division and the number of chromosomes in humans specifically. You might be asked about other different types of organisms and given information about their chromosome number, but we have to know about humans from scratch. The circle just represents a cell. I'm not drawing the nucleus inside, so we need to know that the total number of chromosomes in a human. We're going to talk about the difference between gamete cells, which have the haploid number of chromosomes. We'll discuss that in a moment. Any cell which is not a gamete is referred to as a somatic cell. So your normal, if you like, cells. Anything that's not a gamete is referred to as a somatic cell. So somatic cells in humans have got 46 chromosomes. So the importance of mitosis cell division is that when that parent cell undergoes mitosis and it produces two what are referred to as daughter cells. So once mitosis cell division has occurred, you go from one parent cell to two daughter cells. Each of those daughter cells are genetically identical to each other and the parent cell that it originated from. So they will both contain 46 chromosomes, and they are genetically identical in every way. The sorts of things that mitosis cell division is used for is growth initially. So once fertilization takes place, you start off with a single cell, and in order for that human to grow, mitosis cell division occurs. Whenever you consider and think about growth, growth in questions, it's mitosis cell division that's taking place. Tissue can get damaged for a number of different ways or reasons, and most types of tissue, there are exceptions to the rule, so nervous tissue can't do mitosis cell division, for example, once that nervous tissue has been formed, but most tissue can undergo mitosis to repair when it's become damaged. The other use of mitosis is that some organisms are able to reproduce by doing mitosis, and we'd refer to that as asexual reproduction. Humans obviously can't do that. The other thing that we need to think about, and phrases, terminology we have to be able to use now and moving forward, is haploid and diploid. Haploid can be represented just as the letter N, and really a better word to use instead of haploid, but we can't, is monoploid. So if you think about ploid is referring to the type of chromosome, then monoploid would mean that it's referring to the number of each different type of chromosome. So you have one type of each different chromosome. But that's what haploid is referring to, and it's why N is used. It's the number of different chromosomes that an organism contains. So, for example, in humans, we have 23 different chromosomes. That's how many different ones there are, with different genes within those different chromosomes. In humans and in mammals, the only cells that are haploid and just contain one copy of each different type of chromosome are in gametes, or in the sex cells, or in the sperm and the ova. When we move forward and start to look at meiosis, cell division, in another topic, we'll see that some organisms have very unusual life cycles where actually organisms can exist with the haploid number of chromosomes. And if you think about it logically, as long as an organism has got one copy of each chromosome and they have genes to carry out protein synthesis, then that's fine. But we'll discuss that later. Therefore, diploid, and we've talked about the prefix di before, di meaning two. Diploid therefore means this is where a cell contains two copies of each type of chromosome, and so that can be written as 2n, 2 times n, 2 times the number of different chromosomes. In humans, therefore, that's 46. So a somatic cell, all somatic cells in humans, in mammals, are diploid. Now, one of the important things I think we need to just remember as well, and this is more relevant when we start to look at meiosis cell division, is that diploid is referring to a cell which has got two copies of each chromosome. And so a cell is only diploid if the chromosomes are in homologous pairs. Now again, the relevance of that doesn't need to be talked about now, it's more when we look at a particular stage of meiosis a bit later on. I'm not going to look at the different stages of mitosis in humans, because that would involve drawing out 46 chromosomes, which is just a bit of a faff. So let's think of a different type of organism. So let's say that we've got a cell here. Again, I'm not drawing in the nucleus. The chromosomes are in the nucleus, and this is a somatic cell of this particular organism. If I said that this organism has got a diploid number of 4, you could pause the video and hopefully you could have a go at drawing that. So if we're going to have a diploid number of 4, we need to remember that we've got a total of 4 chromosomes, but they exist in homologous pairs. So let's say we have one chromosome here, another one here. What does homologous mean? It means that you've got chromosomes that are the same length, and realistically most questions are going to ask you to identify homologous chromosomes based purely upon their length. But homologous pairs of chromosomes are the same length, they have the same genes in the same gene locus. But we can't see that based purely on this. And therefore we might have another homologous pair of chromosomes. Again, they're the same length, they're going to have the same genes at the same gene locus. So when mitosis, cell division, occurs, each of the daughter cells is going to contain the same number of chromosomes, and they are genetically identical to each other and to the parent cell which it arose from. Now, this begs the question then, if we're going to produce genetically identical daughter cells, and they contain the exact same number of chromosomes. The only way in which that can happen is if the chromosomes undergo replication. And we've discussed DNA semi-conservative replication and the details of that, and we're not going to look at those details now. But we know that replication must occur. This then leads us on to appreciating that as soon as a cell has been formed, so if we look at our example just here with our organism which has got diploid number 4, as soon as a cell has been formed, it enters what's referred to as its cell cycle. Now I'm going to represent this cell that's just been formed, its cell cycle, as a pie chart effectively. So let's say at this point just here in time, so if we look at this as we move around the pie chart, around the circle, time is elapsing. If the cell is formed at this point just here, the cell spends most of its cycle, so here is its cell cycle, it spends most of its time in what's referred to as interphase. So the vast majority of a cell's life is spent within interphase. Let's label that just on here. And whilst the cell is in interphase, it's just carrying out its function. So if it was a cell in a salivary gland, the primary function of that cell is going to be synthesizing and secreting amylase. Or if you were to look at a particular tissue within your pancreas, those cells there, it might be that during interphase, what its main function is to synthesize insulin, hormone, and secrete it. So the vast majority of a cell in its cell cycle exists during interphase. As the cell starts to move through, as it moves through interphase, and as it's preparing for cell division, whether it's mitosis or meiosis for that matter, but as that cell is preparing for mitosis cell division, it needs to carry out a variety of functions, of tasks, to prepare itself for that division. So a cell only does cell division as part of its cell cycle for a very small proportion of that time. So in this case we are thinking about mitosis cell division. So as the cell starts to come towards late interphase, it has to carry out a variety of tasks, if you like, to get ready for cell division, in this case mitosis. As we're going to look at in a bit more detail in a moment, at the end of late interphase, the DNA is going to replicate itself. It's going to undergo semi-conservative replication, so that each of these two new daughter cells is going to have the requisite number of chromosomes. Cell division, mitosis, is very active. It requires a lot of energy in the form of ATP, so a lot of ATP is going to be synthesized. Organelles also need to be able to replicate, so that the two new daughter cells have got the necessary number of mitochondria or chloroplasts if it's a plant cell. Lots of proteins need to be synthesized, so all of this is going on in late interphase. Now if we have a look at the detail of semi-conservative replication, you can't actually see this. When we look at mitosis in more detail in a moment, remember that during interphase you cannot see individual chromosomes. The DNA is not tightly wound around the histone proteins within the cytoplasm of the cell itself, and it exists as chromatin. You can't see what I'm about to draw actually happening. but it is happening, and it's important to understand that. So I'm going to look at our example of where we've got a diploid number of 4. So let's say that we've got we've got a homologous pair of chromosomes like so, and we have another homologous pair of chromosomes here also. What's going to happen then during interphase is that semi-conservative replication is going to take place. Now if I label these A, B, C and D, so chromosome A will undergo semi-conservative replication, and the chromosome therefore copies itself. The copy of chromosome A is held attached via this structure, which is called a centromere. And what's important is that this is happening with each of the chromosomes. So its homologous pair, that will undergo semi-conservative replication. So we've got A, we've got C. B will do the same. And we can see that it's larger like so. And D will do the same as well. So you can't see this happening in interphase because actually the DNA exists as chromatin. Now, what's really important is that we get our terminology right. We are going to call each of these structures formed, so that whole thing now is a chromosome. So this is a chromosome. Now, really, it's two chromosomes, but we're calling this whole structure a chromosome, and it comprises of now 2 what I call sister chromatids. Why are we referring to this as a chromosome? It's one of the reasons is just to show that we understand that semi-conservative replication has taken place during interphase. And again, this is more important for meiosis cell division that we'll look at a bit later on. So that serves as an overview of the importance of mitosis cell division, but also the cell cycle itself.